All right, it's my pleasure to introduce Matthew Scott. He's a master's student working in the Department of Wildlife, Fisheries, and Aquaculture at Mississippi State University under the guidance of Dr. Mike Sandell, with interests in One Health in aquatic systems. Take it away, Matthew.
Okay, thank you for that introduction. Hello, everyone. Thank you for joining my talk today over the internal microbiomes of Amazonian fishes. So a little bit about microbiomes. Microbiomes are bacterial communities of fishes. I'm specifically speaking today about internal microbiomes. Microbiomes are known to influence things like the overall health of fishes in the sense of like nutrient absorption, which can affect growth rates as well as their immunity. And that's very interesting to different fields like aquaculture and fish farming for purposes of like understanding their bacterial communities and manipulating them to help growth rates of fishes and overall health of fishes when they are trying to get better fish products. But for me personally, I am very interested in the overall ecological understanding of bacterial communities and fishes and how different ecological factors affect the bacterial communities on the inside of fishes. So I'm very happy that I got to be a part of this project looking for tropical diseases and understanding how the bacterial communities in one of the most biodiverse ecosystems in the world are shaped by fishes and different ecological factors. So looking at a few different questions here. So what I'm really interested in is how host taxonomic factors as well as environment and habitat affect bacterial communities on the inside of fishes. And a few studies in the past have asked similar questions with different animals. So looking at this study here, we see that if you transplant the bacterial communities in the guts between a mouse and a fish, we see that the bacterial communities after being transplanted will revert back to the original bacterial community composition of the host. And that shows that the host taxonomic factors really help influence the bacterial community composition on the insides of animals. And looking at this study here, we see that travel in humans, about 2 weeks after travel, that the proportions of bacteria that are heavily associated with dysbiosis, the proportions of those bacteria change as recently as 2 weeks after travel, as well as the proportions of bacteria that are associated with healthy bacterial communities change and decrease. And similarly, after returning from travel, the proportions of those dysbiotic and healthy bacteria revert back to normal after travel. So that kind of shows that locality and habitat can influence the bacterial communities on the inside of animals. And I'm very interested to see how those questions come into play with fishes in the Amazon. So as I said, my fishes that I study are based off of fishes that were collected in this project looking for reservoirs for human disease. And we did our surveys in French Guiana between July 21 and July 2021 and July 2022. We sampled across 3 different seasons, and we sampled across the headwaters, middle tributaries, and coastal lowlands of 3 different main watersheds, as well as along the coast of the country. We sampled along the Mana River, the Santa Maria River, and the Aparog River. And all the data that I'm going to be showing you guys today from fish microbiomes will be from samples that we currently have microbial data on from mainly the Aparog River, as well as a few coastal sites and the coastal lowlands of the Santa Maria rivers and the Mono rivers. So for our field collections for fishes to study, we used dip nets, and we collected each fish individually into 2 milliliter screw cap tubes with 95% ethanol in it to preserve the bacterial communities and as well as the fish tissue. And at each site, we collected habitat water quality using a YSI. And as I said today, I'm going to be showing you guys analysis based off of available bacterial community data that we have. I'm going to be showing you data from a total of 256 fishes that span across 8 different fish families, and from all sample years 2021 through 2022. So to collect the bacterial communities of our fishes, first we had to dissect each individual fish. These fish are about 1 inch long, and we did dissections of the body cavity, and we removed the entire internal viscera of each fish after opening up the body cavity, and we used traditional methods to extract DNA from the internal viscera of each body cavity from fishes. And after we did our DNA extractions, we used PCR to amplify 16S RNA genes in bacteria. And then after we checked the quality of our PCR, we send our PCRs off to Michigan State University Sequencing Corps to sequence our data using Illumina HiSeq sequencing technology. And once we get those sequence data back from Michigan State, we use QIIME2 to give us OTU tables and taxonomic tables in QZA format so that we could analyze our data. So I'm going to go ahead and show you guys some of those comparisons that we did, starting with some of the taxonomic comparisons between fish taxonomic groups. So you can see here, starting with a higher fish taxonomic level, we have alpha diversity, or richness of bacteria, in 2 fish superorders, Acanthamorpha and Austereophyce. We see that there is a significant difference between the species richness or alpha diversity in these 2 superorders. And we also see that Proteobacteria, Firmicutes, and Cyanobacteria are the dominant top 3 dominant bacterial phyla. It's important to note here, make sure with every analysis that we do, you check the R-squared values, because the R-squared values for some of these analysis may be really low, showing that it only explains a small bit of the variation between comparisons. So here we only have an R-squared square value of 0.01, so about 1% of the variation explained here, looking at fish superorders. But if we look at a lower taxonomic level with fish family, we also see a similar result. We have a very low p-value, showing that there are significant differences between the alpha diversity or richness between fish families. And a slightly more bit of the variation here is explained by using a lower level fish taxonomic comparison. And here we can go on to beta diversity. Looking at the beta diversity of the higher fish taxonomic level, Acanthamorpha, we see that there is significant differences in the community composition between bacteria and all of these different fish superorders here, regardless of what level of bacterial taxonomic level you compare with. So we see that we have very significant differences, but like I said earlier, the R-squared value is pretty low, so not all of the differences in these 2 superorders can be explained by just comparing with fish taxonomic levels. And we see the same thing with fish taxonomic levels of host family. We have very significant differences in the bacterial community composition, but only about 5% of the variation can be explained just by looking at the fish taxonomy. So now we're going to get into some comparisons between the locality of the fish, like where it was collected at, and fish bacterial diversity. So looking at alpha diversity between watersheds, so between the Ocrawauk, Mana, and Santa Maria rivers, and then all of our coastal sites, we see that the alpha diversity or richness of the bacteria was significantly different between each one of our sites. But once again, with watersheds as well, we do not see all of the variation that being explained by just comparing where the fish was collected. So looking at the species richness compared between the watersheds or where the fish was collected, we also see significant differences in the bacterial community composition and lower and a very low level of the richness is simply explained by just saying, okay, this is where the fish was collected from. But if we look at some of the water quality data that we collected and compare species richness between different levels of pH, starting with pH, we see that there is very significant differences between bacterial community composition and levels of pH. We see that most of the community composition fell between about 6.5 and 4 on the pH scale, and that 33% of our variation could be explained by looking at pH. So a lot more variation is explained by looking at some of these water quality parameters. So let's look at temperature. So temperature explained 42% of our variation in bacterial community composition. And we saw a very significant difference in the bacterial community composition between fishes that were caught in different temperature water. So we can see that different water quality measures may be more useful for comparing bacterial community composition. So in conclusion, different levels of fish taxonomic comparisons do show that different fish taxonomic groups have different community or bacterial diversity, but they don't explain the entire, they don't fully explain why the bacterial diversities are different. And the same with like localities of the fishes. We do see a significant difference in the bacterial diversity between sites where the fish were collected, but that's not simply explained by the locality. We see much more of the variation explained by some of the water quality that we collected, like pH and temperature. Some of our future goals here are going to be to try to identify factors that are some of the main factors that shape the fish microbiomes in our study area, and to try to characterize the diversity of fish microbiomes in the Amazon, and as well as try to see if we can find any fishes that are positive for containing Mycobacterium ulcerans, to try to look into seeing whether or not these fishes are possible reservoirs for the disease. These are my sources for this presentation. I'd like to thank my lab mates, David Pounders, Mason Strickland, Zach Alley, Alex Rakestraw, NSF for funding, the NACA, and all of the Mississippi State students and faculty for their support on my project. So thank you guys for listening, and I'll be willing to answer any questions that you guys might have.
Thank you very much. That was an excellent presentation and an amazing study site.
Thank you.
Folks, if you got questions, feel free to unmute yourself or drop them into the chat. Okay, we got a few coming into the chat here. You got getting lots of compliments here too. They're curious if you reared these different fish in common source water and fed similar diets, you think you'd still see the level of variation. Basically, is it the fish physiology, or is it driven by environment, diet, and what implications, if any, does that have in the application of probiotics?
We haven't done any comparisons between our fish's diets. We do have data on like the macro invertebrates that were collected at each different site, so we may have an idea of what the fish could be eating, but these fish were very small, and we would have to use some genomics tools to see what type of food that they were eating. But we have not tried those comparisons yet, but that would be something that we'd be interested in looking into if our methods allow us in the future.
Great. Another question here. were you targeting any particular phyla or species?
At first we weren't, but as I got further into my thesis, I wanted to get like a better assay for looking at different comparisons with, especially with like fish taxonomy. So we started comparing fish specifically in families Labiacinidae and Characidae to try to compare between lower level fish taxonomy groups. And we're also going to try to use genomic tools to try to identify some of those fish down to species to identify different bacterial diversity between a lower level of fish taxonomy. So yes, we did try to target certain fish taxonomy groups.
Great. And one last question here. Do you think that using the dip nets could have biased the sampling in any way? They think it's possible that less healthy fish would be easier to catch with a dip net, and that might influence the microbiota.
I feel like it might have biased the sampling just based on some of the habitats where the fish were collected. I feel like some fish may have been like in deeper water, and we may not have been able to get like as even as a sampling of the rivers as we could have. But I feel like we also are collecting fish from like similar habitats in the river system. So I feel like that would be better for doing comparisons between fish bacterial communities. But I feel like there are definitely ways that we could get more even species richness throughout the river habitats. That
That
kind of
sounds
answers the
good.
question.
Sounds like you need to go back down to French Guiana there, Matthew. Continue the
Exactly.
great work. Thanks a lot for the presentation.
Thank you.